Hi everyone. Today we are discussing about the physiology of gastrointestinal system. In this lecture, we are expected to differentiate between mechanical and chemical digestion. We'll be describing the composition and functions of saliva. You should be able to list out the functions of stomach. You should be able to explain the phases of gastric secretion, discussing the functions of liver and pancreas. We'll be discussing the factors involved in controlling biliary and pancreatic secretions. We'll be describing the significance of bile in digestion of fats. We'll be describing digestion and absorption in the small intestine, and towards the end we'll be summarizing the role of large intestine and the events involved in defecation reflex. Let us now see the functions of digestive system. Mechanical breakdown of food in the oral cavity. When we consume the food is broken down into several smaller fragments by the grinding action of the teeth. The second action is motility, which involves the mixing and movement of the food contents in the forward direction in the digestive system. There will be several secretions which is from the digestive glands, such as enzymes, hydrochloric acid, and various digestive juices added into the foodstuff. Digestion, the chemical process by which the foodstuffs are acted upon by the digestive enzymes. And absorption, which is necessary nutrients are reabsorbed into the bloodstream. And this is the diagrammatic representation of the events happening in the digestive system. You can see here when the food is acted upon by the various digestive enzymes, it is broken down in the gastrointestinal system. The motility is the process by which it is moving forward in the forward direction. Absorption, the necessary substances will be reabsorbed into the blood, and the unwanted substances will be excreted or eliminated out. That will be at the end of the gastrointestinal system. There will be secretions, which is from the various digestive glands situated throughout the gastrointestinal system. It will be added into the foodstuff, and then it will be moving forward. Motility will be taking it moving forward. Gastrointestinal processes. Ingestion is the term which is used when the food is placed in the mouth, right? So that is a voluntary process. Propulsion, movement of food through the gastrointestinal tract, which involves swallowing, which is a voluntary process, and peristalsis, which is an involuntary process, which includes the movement of food in the forward direction. Digestion is the mechanical breakdown of the food, which includes chewing in the mouth, which is termed as mastication. Mastication, churning in the stomach, which includes a segmental contraction. So stomach is also having a role in the mechanical breakdown of the food materials of the food bolus when it reaches the stomach. Chemical digestion of the food. Large food molecules are broken down into chemical components by the action of the various digestive enzymes. Absorption. The products of the digestion are transported from the lumen of the gastrointestinal tract to the blood and the lymph. At the end, the defecation process is the elimination of indigested product via the anus as feces. So that is how it is excreted out from the body. Let us now learn about motility. Peristalsis, alternating waves of contractions and relaxation of the muscle in the adjacent segments of the organ wall, squeezes the food along the tract. Churning movement, which is the grinding movement, especially it is seen in the stomach, which will be again breaking down into larger foodstuffs are broken down into smaller one. Segmentation, rhythmic local constrictions will be seen. So there will be continuous areas of constrictions and relaxations. So this will make the food into this will make the food into smaller fragments that will be occurring in the intestine. It mixes the food. The food here, you can see here, it is called as chyme. Chyme is the term which is a semi-solid food. When the food is ingested into the mouth, that will be converted later into bolus, and when it is into the digestive tract, when it is mixed with various digestive enzymes, this will become a semi-solid mass, which we call it as a chyme. Chyme is the semi-solid mass of the food. When it is mixed with the digestive juices, then it exposes the food or the chyme to the absorbing surface. Mass movements, strong peristaltic waves that push the contents of the colon towards the rectum. So usually the mass peristalsis is seen in the colon, which is the large intestine. Let us now learn about the peristalsis, the process by which the food bolus is moving forward in the gastrointestinal system. You can see that the food bolus is located here, but the front area is relaxed, and behind the food bolus, that area is constricted, which will cause a pressure change, which will be pushing the food in the forward direction. Right? This is a high pressure system, and this will be a low pressure system. So always there will be a movement which is taking place from high pressure to low pressure areas. So food bolus will keep on moving like this because these constrictions will be a kind of movement how you see here. Right? You can appreciate how this movement is taking place here. So the constriction will be taking place, and then it will make the food bolus to move in the forward direction. The propulsion of food in the forward direction, this is called as a peristalsis. There is another type of contraction which is called as a segmentation contraction, which usually it is seen in the small intestinal segments. So you can see that the segmentation contractions, the whole bulk of the bolus, there will be in the intestine, there will be alternate relaxation and constrictions. These are the constricted segments and these are the relaxation segments. In the next few seconds, you can see that it alters. This part which was relaxed, that has now become constricted. And here you see that this part which was constricted, it has become relaxed. So the alternating contraction and relaxation of the parts of the skeleton, the smooth muscles within the small intestine will bring about mixing of the food. So the large food materials will be broken down into smaller segments, and this is one of the important function of the small intestine. So here also you can see how the contraction is taking place. So this is a kind of contractions which is very commonly seen in the small intestinal segments, which is called as a segmentation contractions. Let us now see the control of the gastrointestinal tract. The motility and secretion are regulated, which can be increased or decreased depending upon the physiological need of the body. The four major factors will regulate the gastrointestinal functions. Autonomous smooth muscle functions. They are specialized muscle cells that display rhythmic, spontaneous variations in the membrane potential and are self-excitable. They are called as interstitial cells of Cajal. This is termed as Cajal, K-A-J-A-L. It's not pronounced as Kajal. It is Cajal, interstitial cells of Cajal. So that is how it is pronounced, and that is a special type of cells which is displaying the autorhythmic kind of property. That means if a small piece of the intestine is cut and if it is kept outside, it will keep on contracting, right? So this is one thing which you will be seeing it if you are doing it on animal experiments like a rabbit or a kind of a rat or whatever smaller animals if you take, and if you are doing a dissection of the gastrointestinal system, and if you keep it one piece of this cut end, if you keep it in a petri dish, you can see that that will be contracting because of this kind of autorhythmic cells, which will be having variations in the membrane potential. Intrinsic nerve plexus is located, which is the local nervous system of the gastrointestinal tract. This is a very important one. This is a local nervous system. It's the intrinsic nerve plexus. Within the gastrointestinal system, there is a kind of nervous system which is called as an intrinsic nerve plexus. The alimentary canal wall consists of two important networks, which includes myenteric plexus and submucosal plexus, right? So this will be having myenteric plexus and submucosal plexus, which is giving rise to this particular type of a local nervous system, which is called as an intrinsic nerve plexus, which will be influenced by other factors. And there is extrinsic nervous system, which are already the branches of autonomic nervous system, which includes the parasympathetic and sympathetic. And here you need to remember that parasympathetic will be having a stimulatory role in the gastrointestinal system, and sympathetic will be having an inhibitory role on the gastrointestinal system. So it is, in brief, you can think that it is the gastrointestinal system is highly influenced by the parasympathetic nervous system, because that is the stimulatory influence on the gastrointestinal system. And there is also gastrointestinal hormones like gastrin, secretin, cholecystokinin. So this particular chart will give you the overview of how the gastrointestinal system is controlled. You can see that there is intrinsic nerve plexus, extrinsic autonomic control, which include the sympathetic and parasympathetic. And I mentioned that the parasympathetic is having the predominant role here, because parasympathetic is the stimulatory effect. And also the gastrointestinal various hormones we'll be going through. Physiology of digestion in mouth. The teeth mechanically break down the food. Mastigation that involves the slicing, tearing, grinding, and mixing of ingested food by the teeth. That process is called as mastigation. It begins the carbohydrate digestion. One thing you need to remember is that the food stuff is not remaining inside the mouth for a longer duration. As soon as we take the food in our mouth, it will be chewed and it will be swallowed. So we don't have much of time that the food stuff will not be spending much of time inside the mouth. So a small amount of carbohydrate digestion will begin in the mouth. Secretion of saliva. So saliva will be mixed with the food. Saliva is made up of 99.5% of water and 0.5% of electrolyte. Means. Let us see the salivary secretion. Salivary secretion is continuous and can be increased by reflex. There are two types of reflexes which we need to remember. It is the simple and conditioned salivary reflexes. Autonomic influence on salivary secretion is very important. Digestion in the mouth is minimal and no absorption of nutrients will occur. So here, as I mentioned, the food will not remain in the mouth for a longer duration. As a result, only the carbohydrate digestion will just begin, because we have the salivary amylase in the saliva, which will be starting or initiating the carbohydrate digestion in the outer part of the bolus. By the time it will be already swallowed, then it goes to the next part of to the stomach, right? It will be going through the esophagus and then it will be going to the stomach. So the food is not staying much time in the mouth. Digestion in the mouth involves the hydrolysis of polysaccharides by salivary amylase. Most digestion occurs after swallowing and some in the stomach, mostly in the small intestine. So majority of the digestion process or the major part is taking place in the small intestine. Very little is taking place in the stomach, but the least will be happening inside the mouth, as the food will be staying only for a shorter period of time. So you can see that it is produced by the saliva, is produced by 3 salivary glands as mentioned in this picture. Approximate secretion is about 1500 ml, and the contribution is by the 3 salivary glands. The submandibular salivary gland, which contributes to maximum, which is 70% of saliva, is contributed by the submandibular salivary gland. Then we have 20% by the parotid gland, and we have 5% by the sublingual gland. We shall now discuss on the regulation of salivary gland secretion. This is a very important slide to understand. We need to remember that there is a center called salivary center, which is located in the medulla. When I'm talking about every center, finally it comes to the medulla, right? We discussed about the cardiovascular center, we discussed about the respiratory center. So remember that medulla is a highly sensitive center which lodges many of the vital centers. So in this case of digestion, there is a center called salivary center which is located in the medulla. When you stimulate the salivary center, the salivation will happen, right? That will be the. Now look at these 2 reflexes. The first one we talk about is the simple reflex. There are 2 reflexes here, simple reflex and conditioned reflex. Let us first talk about the simple reflex. When the food is placed inside the mouth, remember, when the food is placed inside the mouth, the pressure receptors and the chemoreceptors in the mouth will be sensitive then. When food is placed inside the mouth, the pressure receptors will sense a kind of pressure, because inside the mouth it is pressing against the receptors, right? It will be exerting a pressure. Chemoreceptors, we have got taste buds on the tongue and around the mouth, so that will be all around. You'll be seeing that it will be sensing the amount of chemical substance in the food. So these are the stimulus which will be then sending the information to the salivary center in the medulla. And salivary center, through the autonomic nerves, it will be sending the information to the salivary glands to bring about salivation. So salivation will happen. So this is a kind of a mechanical event where you are just placing the food in the mouth, and then it will be stimulating these receptors, and then it will be causing the salivation, right? Salivary secretion. So that's a simple reflex, right? So this happens in every individual. What about a conditioned reflex? Conditioned reflex requires higher brain control, like cerebral cortex, right? Here are the examples. Thinking about the food, seeing the food, or smelling the food. Any of these 3 stimulus, it might stimulate the cerebral cortex. It will stimulate the cerebral cortex, and cerebral cortex will be sending the information to the salivary center, and then through autonomic nervous system, the person will undergo salivation. Conditioned reflex requires the prior knowledge about the food. Now just compare an infant with an adult. We all know about different food stuff, and you might be having some food as your favorite food items, right? So if you are very hungry, and if your mom prepares one of your favorite food item and kept there, the smell of the food, or when you see the food, or when you are thinking about the food, any of these stimulus will stimulate your cerebral cortex, and your salivary centers in the medulla will be stimulated. So this is a kind of a conditioning. You should know, or you have a prior knowledge about the food. But what about in the case of an infant? They don't know anything about the food. So a conditioned reflex may not function in the case of a newborn baby or an infant who doesn't know anything about that food. In them, the simple reflex will be very active. When you keep the food in their mouth, they will start salivating. but they don't know how any other item looks like, right? Like if you are fond of albake, right? When you see the albake, when you are in a hungry situation, when you didn't had your lunch, your breakfast, and by 3 p.m. or 2 p.m. somebody has brought albake, then what will happen? You will start salivating. Automatically you will start salivating because you know about the taste and you are very hungry. But at the same time, if it is shown in front of a newborn baby or an infant, they doesn't know how it looks like or how it tastes like. So unless it is tasted, or the prior knowledge is very much required for the conditioned reflex to occur. So this is the difference between the simple reflex and a conditioned reflex. So in the human beings, in an adult human beings, conditioned reflex is very, very strong, right? So that is important. Let us now see the functions of saliva. Helps in the perception of taste. The food materials will have to first get dissolved in the saliva, then it has to be presented to the taste buds for the perception of taste. For the formation of bolus, when you take the food materials in the mouth, it finally mixes with the saliva and it forms a ball like structure, and that is called as a bolus, a food bolus. And it is the food bolus which will be then swallowed into the esophagus and it will be moving forward. The digestive function in saliva, the salivary amylase, which will be acting upon the cooked starch. As I mentioned before also, it is only a very small part of the bolus which will be digested. So the digestion just begins in the mouth, right? So because the food is not spending for a longer duration inside the mouth. It keeps the oral cavity moist. It is necessary for articulation or for the speech. This is very important because if somebody is not having the salivation, right, so he cannot speak. You will become speechless, right? If you hear a sad news, if too much of like activity of the autonomic nervous system which will stop the salivary secretion and you may not be able to speak. So saliva is necessary for articulation or for the speech. Protective function provides oral hygiene as it contains lysozymes which is having bactericidal action. Now let us discuss about the deglutition or swallowing. The swallowing begins as a voluntary activity after the food bolus is formed in the mouth. It includes three phases: the oral phase, which is voluntary, pharyngeal phase and esophageal phase are involuntary. Remember, so these two phases, the phases 2 and 3, which is pharyngeal and esophageal phase, these are involuntary, which means they cannot be stopped according to our will. Now let us compare the oral phase and the pharyngeal phase of swallowing. You can see the green, this is the food bolus, and in this particular part you can see the epiglottis can be seen. What is epiglottis? Epiglottis is the structure which will be closing the glottis and that will be preventing the food from entering into the respiratory tract. So in this picture you can see that the epiglottis is closed so that it will be preventing the food to enter into the respiratory tract. So the food will be going from this direction towards the digestive tract, right? So that will be the right pathway. So here it was just up and you can see that epiglottis is glottis is here and this is the epiglottis is up. So here upper esophageal sphincter contracted and epiglottis is about to close. So in the second phase you are seeing that the epiglottis is closed so that it will be preventing the food from entering into this particular lungs, right? So the respiratory tract. So it will be taking this route towards the esophagus. And esophageal sphincter is relaxed. The upper esophageal sphincter is relaxed so that will be allowing the movement of the food bolus into the esophagus. But here you can see that in the first phase it is in a contracted state. because the food is still here. It has to go all the way here. By the time it reaches here, this will be relaxed. So esophageal sphincter. So here it is controlled. So that's the difference. In the first phase, you will see the esophageal sphincter contracted. In the second phase, the esophageal sphincter is relaxed and glottis is also closed. So this is how it will be taking place. You can see the food is then forwarding. As soon as the food enters, you can see that the esophageal sphincter is again contracted, right? So the food, so it will be just allowing the food. So as soon as the food bolus goes to the next part, the upper esophageal sphincter will be contracted. So you will be see this wave of relaxations which will be propelling the food in the forward direction. So the peristalsis is taking place, the muscles are contracting in between, and then relaxation waves are also seen. Finally, it reaches the stomach. So the upper esophageal sphincter contracted and epiglottis is up. So here also you can see that after this is moved, epiglottis is going back to the normal location. And this is called as this kind of reflex is called as a deglutition reflex. A deglutition reflex, which means a temporary stoppage of respiration or temporary stoppage of breathing when the person is swallowing. See, when the epiglottis is closed like this, you cannot take or you cannot breathe air because this is closed, right? So the air will not be entering from the nose. This will be closed, right? So there is a temporary stage during the time of swallowing where the respiration stops. This is called as a deglutition apnea. Deglutition apnea. Apnea means it's a temporary cessation of breathing. So it's breathing, stoppage of breathing or momentary stoppage of breathing as a result of deglutition or swallowing. That is called as deglutition apnea, right? So there is a deglutition center which is located again in the medulla, which will be helping in this particular reflex. Deglutition center, same like in the salivary secretion, the salivary center, there is a deglutition center which will be also located in the medulla, which is responsible for this action. So please remember that during the deglutition, there is a momentary stoppage of breathing. What will happen if this doesn't work? When you cannot breathe and swallow at the same time. If that happens, you might choke. The food materials might enter into this, right? So if it is happening like this, the food materials, instead of going into this part, instead of going into the esophagus, it might enter into the respiratory tract. That is the reason why the epiglottis is always closed. There is a temporary stoppage of breathing. So this is essential to know. And that is the esophageal phase, which is the last phase. So it has got three phases: oral phase, pharyngeal phase, and the esophageal phase when the food is in the esophagus. Just have to differentiate all the three phases. Now let us see the functions of stomach. Stomach acts as a reservoir function, which means it temporarily stores the food. Gastric filling involves the receptive relaxation, which will allow the stomach to accommodate the meal. This reflex is initiated by vagus nerve. So when the vagus nerve is active, this will cause a kind of relaxation of the stomach muscles, receptive relaxation. This is the reason why the pressure within the stomach will not increase too much when we consume the food. See, till we are full, we can keep on eating. Otherwise, a small chocolate or a small something very small, when you take, the pressure might increase within the stomach as soon as it enters the stomach, right? So we will not be able to accommodate more amount of food into the stomach because of this receptive relaxation, which is mediated by the vagus nerve. It will help us to accommodate more food. Secretory function. The secretion will be by the gastric. The main secretion is made in the stomach is by the gastric juice, which contains the HCl, which is the hydrochloric acid, then pepsin, which plays an important role in the protein digestion. So the protein digestion will be beginning in the stomach. Then there is a protective function. The high acidity in the stomach due to the HCl has a bactericidal effect. It will kill the bacteria which is entering because it is acidic. Because acid substance, the HCl is an acid, so it can kill the bacteria, bactericidal effect. Hemopoietic function. It secretes intrinsic factor which is essential for the absorption of vitamin B12. This is again a very, very important point. When we discussed about the formation of red blood cells in erythropoiesis, we discussed that erythropoiesis requires a nutritional factor. One of the important nutritional factor is vitamin B12, vitamin B12, right? So what for the absorption of vitamin B12 from the intestine, it requires an intrinsic factor which is released from the stomach. So the stomach cells will be releasing this intrinsic factor which is also essential for the reabsorption of vitamin B12, which means if you don't have this intrinsic factor which is released from the stomach, we cannot have reabsorption of vitamin B12. So, shall I tell an example? If a patient with a partial gastrectomy, half of the stomach is removed, probably he is having a gastric carcinoma, right? Half of the stomach is removed. In that case, what will happen is there is a deficiency of the intrinsic factor released from the stomach, and as a result, this patient will not have enough amount of vitamin B12, and then what will be the result? There will be a lack of RBCs. The RBCs which is forming, it is immature. Immature RBCs can be seen. Those kind of situations you will learn later. That condition is called as a megaloblastic anemia. Megaloblastic, that means the size of the RBCs will be larger in size because they are immature, because they lack vitamin B12 as a result of absence of intrinsic factor secreted from stomach. HCl in the stomach is needed for the conversion of ferric iron to ferrous iron, right? This is for the iron. It has to be converted from ferric form to ferrous form before it is reabsorbed, and this conversion is done by the hydrochloric acid. It's a biochemical step there. Now let us see the cells which is involved in the gastric glands and their secretions. These are different types of cells you will be learning. Surface mucus cells which secretes the bicarbonate rich mucus, mucus cells which secretes thin acidic mucus. So here mucus itself is two types, bicarbonate rich mucus and acidic mucus. Then there is a parietal cells which secretes the HCl or hydrochloric acid and intrinsic factor. This is the intrinsic factor which we discussed in one slide before. That means this intrinsic factor is needed for vitamin B12 absorption. So remember, parietal cell has two functions. It will be secreting HCl and the intrinsic factor. Endocrine cells which secretes hormones like gastrin and histamine, then chief cells which will be secreting pepsinogen. So this is the flow chart, will gives you an idea of how it is happening. So you can see from here when the protein will be broken down by peptide to peptides by pepsin, right? So here you can see the chief cells will be secreting pepsinogen. Pepsinogen is converted to pepsin by the action of HCl, right? HCl is again secreted by the parietal cells. So parietal cells is secreting HCl and also the intrinsic factor. And what is the intrinsic factor doing? Intrinsic factor is required for the vitamin B12 reabsorption, right? So that will be happening, vitamin B12 absorption. So these are the functions of various cells. So you need to know what are the functions of each of these cells. And mucus cells usually secretes the mucus. Bicarbonate rich mucus will be protecting the surface of the or the gastric mucosa, because here inside the stomach you know that there is HCl or hydrochloric acid which is acidic. So you need some kind of base to neutralize the acid. Otherwise, the HCl can corrode the gastric mucosa. It can kill or destroy. So in order to protect, we have to have a lining of the bicarbonate rich mucus layer there. So that is the function of that surface mucosal layer. Gastric secretion takes place in various phases. The first phase is called as a cephalic phase. Cephalic, which means it is the head, right? The head region. Activated by mental factors. It's the psychological factors. Vagus nerve stimulates the gastrin in the stomach, which causes the gastric secretion. Now look at this slide. In this phase, when the individual is thinking about the food, sensation or thoughts about the food, it will be stimulating his higher centers, and it will be then through the vagus nerve, through this parasympathetic nerve, these are the vagus nerve, it will be stimulating the stomach mucosa, and then it will be secreting the gastric juice. And this also will be causing a kind of hormone release, which is the gastrin, and then gastrin can bring about the gastric juice, right? So this will be happening. So here, this particular phase is purely neural in origin. That means the sensation, either you can see the smell of food or thought of food, or when you're seeing a food, any of these things will cause mental factor. That's why it is written mental factor. It will be having, the brain is involved, the prior knowledge is required, right? So that is when you see the food, you will be, that time, in the stomach, although you are seeing it, but in the stomach will be prepared to digest that food, so that slowly the gastric secretion will start in the stomach. It is anticipating an entry of a food material into the stomach. So in anticipation, it is just preparing, the stomach is preparing to receive that food, and slowly the gastric secretion is increased in this phase. And this is called as a cephalic phase, which is purely mediated by the vagus nerve. Suppose if you cut the vagus nerve in an experimental animal, if you cut the vagus nerve, this phase, the cephalic phase, will not occur. Why? Because if you cut the vagus nerve, there is no stimulus. Even if the person see the food, this information has to go to the stomach. So these nerves are cut, right? So it is purely vagus in origin. The second phase is called as a gastric phase, where the food has to be present inside the stomach, right? So this is another thing. So the product of the protein digestion in the stomach will be stimulating the release of gastrin, right? So here it is direct distension of the stomach will also stimulate. So here two mechanisms are there. When the food enters into the stomach, this food enters and it will cause distension or stretching of the stomach, right? When it starts stretching, this will be taking an information through the nerves. It is again the vagus nerve. It will be taking the information to the brain, and through the vagus nerve, the information will be coming back, and it will start stimulating the gastric secretion. At the same time, here gastrin is also released as a result of food which is present inside the stomach. So this particular protein digestion which happens, that will be stimulating the release of gastrin. So this is a hormone. So here in this phase, this is partly hormonal and partly neural. So partly hormonal and partly neural, because what is the hormonal part? The hormonal part is the gastrin, and it will be released into the bloodstream, and it will be causing the gastric secretion, right? But in this case, the distension, the mechanical distension of the stomach itself is required, itself will bring about a neural mechanism. So a neural component is there. So this is the efferent and efferent, both will be traveling through the vagus. And sometimes this reflex is also termed as a vago-vagal reflex. Vago-vagal reflex, because efferent and efferent fibers will be going through the vagus nerve. So this you will be reading later In some of the books, when you read about the vagus nerve involvement in the gastric phase, they will mention it as vagovagal reflex, which means the efferent is going, this is the efferent fibers which is going through the vagus, and efferent is coming through the vagus. So it has partly neural and partly hormonal. Now my question is, when you do a vagotomy, when you cut the vagus nerve in an experimental animal, this phase, will it occur or not? Yes, it will occur, but to a lesser extent, because here when you cut this vagus nerve, this vagovagal reflex will not happen, but still this protein digested products will be there, and that will stimulate the gastrin, and gastrin can bring about gastric juice secretion, right? So the amount of gastric juice which is produced might be lesser in quantity, but still it will not stop because it is partly neural and partly hormonal. What about the previous one? The previous one, what we saw is about the cephalic phase, which was purely neural. So that means when you cut the vagus nerve, no information is reaching the stomach and no gastric juice secretion. But here it is partly neural and partly hormonal. So this is what it is important. Now the third phase is called as an intestinal phase. Intestinal phase is that when chyme reaches the duodenum, the fats and carbohydrate and acid contents in the chyme stimulate release of gastric inhibitory peptide, which is called as a GIP, cholecystokinin, which is called as a CCK, and secretin. This will decrease the gastric secretion. Please remember, this phase there is a reduction in the gastric secretion, whereas cephalic phase and gastric phase, we saw that the gastric secretion was increasing. But here it is the intestinal phase, when the food leaves the stomach, right? Here the food leaves the stomach, and when it reaches here, you are seeing that in the stomach, see, once the food leaves the stomach, we don't want the gastric juice to be secreted because no food is present here. What's the point? So the food is already entered into the duodenum, so we don't want the gastric juice to be produced inside the stomach. So this is always an inhibitory phase, so it decreases the secretion of the, so here when the food materials enter, see the chyme, when it reaches the duodenum, the fats and carbohydrate and the acid content in the chyme will stimulate all these hormones, GIP, secretin, CCK, all these will be then inhibiting the gastric secretion. See, it is written decreased secretion. So we don't want gastric juice to be continuously released when we don't have a food material inside the stomach, because then it can lead to gastric ulcers. So we don't want that to happen. So it is the inherent property of this particular reflex that it is an inhibitory phase. Intestinal phase is an inhibitory phase. Pancreatic secretion. When you look at the pancreas has got an endocrine part and an exocrine part. The endocrine part, we already dealt with it during the endocrinology lectures. The islets of Langerhans, which is capable of releasing the hormones insulin and glucagon. In this lecture, we are mainly focusing our view on the exocrine portion, which includes the duct cells and the acinar cells. The duct cells and acinar cells, which will be responsible for the digestive enzyme formation. So let us see the digestive enzymes and the secretions from this. Pancreatic duct cells releases bicarbonate rich secretions. So if you look at the duct cells will be responsible for the release of bicarbonate rich pancreatic secretion, and pancreatic acinar cells that will be responsible for the secretion of digestive enzymes. Pancreatic exocrine secretion is regulated by two major hormones, the secretin and the cholecystokinin, which otherwise called as CCK. So CCK is the standard abbreviation for cholecystokinin. Secretin and cholecystokinin, this is the regulatory enzymes for the pancreatic exocrine pancreas. The pancreatic digestive enzymes includes amylolytic enzymes which acts on the carbohydrates such as pancreatic amylase, lipolytic enzymes that acts on the fats such as pancreatic lipase, proteolytic enzymes that acts on the proteins like trypsinogen, chymotrypsinogen and procarboxypeptidase, each of which are secreted in inactive form, which gets activated when released into the duodenal lumen. Let us now see the regulation of pancreatic secretion. Whenever the acid content in the duodenal lumen enters, it will release the hormone secretin. Secretin is a hormone that will be released from the duodenal mucosa, and that will be secreted into the blood because it's a hormone. It is released into the blood, then it will go to the pancreatic duct, and then it will be stimulating the pancreatic duct cells to secrete the secretion of aqueous bicarbonate solution into the duodenal lumen. So here, in order to compete with this acid, we want some bases. So you can see that here bicarbonate. So this neutralizes the acidic contents what is coming into the duodenum, right? So it's a kind of a neutralization. So remember that the pancreatic duct cells plays an important role here. Like it will be the one which will be secreting the bicarbonate rich secretion, but which is the hormone responsible. It is the secretin which is released from the duodenal mucosa, which is responsible for the stimulation of pancreatic duct cells. Now let us see what happens when fat enters into the duodenal lumen. Fat and protein digested product. It will stimulate another enzyme which is called as CCK, cholecystokinin, which is released from the duodenal mucosa. And again, CCK, because it is a hormone, it is directly released into the bloodstream. Through the bloodstream, it will reach the pancreatic acinar cells, and that will increase the pancreatic digestive enzymes. Here, because the fat and the protein digested products are reaching into the lumen, so we need some enzymes to digest that. So in order to digest that, you are seeing that the pancreatic secretion will be rich in pancreatic digestive enzymes. So pancreatic secretions can be in two types. It can be either basic secretions or bicarbonate rich secretions, or it can be more in digestive enzymes. So this depends upon the situation. So here it has started with arrival of fat and protein digested products. If that such kind of things are arriving in the duodenal lumen, we need that to be digested. So more digestive enzymes are secreted. Here it is the acidic content which is entering into the duodenum. So we need to neutralize the acid. So here basic contents are released, right? Aqueous bicarbonate solutions is released. Let us now learn about the functions of liver. Liver is the major gland where all the metabolisms will be taking place. After the absorption from the intestine, all the metabolic phases take place in the liver. It is involved in the secretion of bile. The synthesis and secretion of bile is the major function of liver. The glycogen, vitamin B12, fat, iron, copper, etc. are stored in the liver. It is a source of plasma proteins. The plasma proteins, what you see in the plasma, is synthesized in the liver. It is having a major role in activation of vitamin D. We have seen that the vitamin D is synthesized and it is activated in different steps. The first step is taking place when it is exposure to sunlight in the skin. The second step in the liver. The third step will be taking place in the kidney. Excretion of cholesterol and bilirubin is another function of the liver. It is having hemopoietic functions in the fetal life, in an adult life, and in the infant. After the birth, you are seeing that it is produced in the bone marrow. The hematopoietic functions, the hemopoietic functions is from the bone marrow, right? The production of red blood cells and the cells will be in the bone marrow. But in the fetal life, you can see that there is a stage where it will be produced from the liver. Before that, there is another stage where it will be produced from the yolk sac. So after the yolk sac, the hematopoietic functions will be then initiated by the liver. Then it will be taken over by the bone marrow, red bone marrow. Detoxification of certain hormones and drugs. Whatever harmful drugs or medications you take, that is finally detoxified in the liver. That is why always the physician says that unnecessary we should not take any medication without a valid reason, because the reason being it is a toxic effect will be seen in the liver, because liver is the source of detoxification of all these drugs and even the hormones. It is detoxified in the liver. Removing bacteria by a special type of cells, they are called as a Kupffer cells. Kupffer cells are phagocytic cells in the liver which is capable of engulfing the bacteria and other debris. So these are some of the important functions of liver. Now let us see the functions of gallbladder. Gallbladder stores bile and it releases bile to the duodenum when it is required. Look at this picture. This is the gallbladder and this is the cystic duct, and you can see that the common bile duct from both ends, you are seeing that these are the duct and it forms the common bile duct. From the hepatic duct it forms a common bile duct. So it is actually the liver which synthesizes, and then it will be coming through this pathway and it will be stored in the gallbladder, and whenever it is needed, it will be gallbladder will be squeezed, and through the cystic duct it will go through the common bile duct, and common bile duct opens to the second part of the duodenum. When you look at the duodenum, it is having three parts. The first part, second part, and third part is like appearing like a C-shaped. So the head of the pancreas will be within this C shape. So you can see that this is the common bile duct. The common bile duct and pancreatic duct joined together, and it will be opening together into the second part of the duodenum. So that is how it is secreted. Bile is secreted. So gallbladder is having an important function in storage and concentration of bile within the gallbladder. Acidification of bile is another function of gallbladder. Concentration of bile received from the liver. So initially, whatever bile it receives from the liver, it is very dilute, but it will be highly concentrated before it is released into the second part of duodenum. This also regulates the pressure in the biliary system by contraction and dilation. So here the gallbladder is capable of contraction, and then it can maintain the pressure within the biliary system. So that is also one of the functions of the gallbladder. Let us see the significance of bile in digestion of fat. Bile salts aid fat digestion and absorption. It is a kind of a detergent action. That means larger fat droplets will be broken down into smaller fat droplets. That is the detergent action. When the particle size decreases in size, it will be easier for the enzymes to act. Remember one thing, the bile is not a digestive enzyme. Bile, once it is secreted, it is only function is that it is having a detergent action. That means the larger fat droplets, it will be emulsifying into smaller fat droplets. That process is called as an emulsification of fat. So it will be made into smaller fat droplets, then it will be acted by the pancreatic lipases. Formation of micelles. You can see that micelles, this is the structure of a micelle. Bile salts and lecithin aggregate in small clusters with their fat soluble parts huddled together in the middle to form a hydrophobic water fearing core. So that will be in this way. You can see that it is all joined together. Right? So that is a hydrophobic water-fearing core, and that is how the micelles are formed. Bile salts stimulate bile secretion. Cholecystokinin, CCK is the cholecystokinin, that's the hormone. Cholecystokinin promotes the gallbladder emptying because it causes contraction of the gallbladder emptying, and this function is sometimes referred as cholerectic. Any substance that increases the bile secretion, cholerectic substance, right? So it will be increasing, CCK will be promoting gallbladder emptying. So that is one of the function. Bilirubin is a waste product excreted in the bile, and it does not play any role in digestion. So this is an important one. It is not a hormone, or it is not an enzyme. It's the only function what bile does is it will be breaking down the larger droplets of fat into smaller one, and that will be acted upon by various digestive enzymes like pancreatic lipases, which will be digested then. So it doesn't have any role in the digestion as such. Let us see the action of hormone cholecystokinin on gallbladder. When the protein digested product and the fat reaches the duodenum, it will be secreting the plasma cholecystokinin, CCK. And cholecystokinin acts on the gallbladder, which brings about contraction of the gallbladder. So you can see that gallbladder, once it contracts, the bile will be moving into the second part of duodenum. Entry of bile will be taking place. At the same time, cholecystokinin also acts on the sphincter of Oddi. Here there is a sphincter of Oddi, where there is an opening between the pancreatic duct and the bile duct, sphincter of Oddi. So sphincter of Oddi is brought about relaxation. Like whenever cholecystokinin acts, it contracts the gallbladder, it relaxes the sphincter of Oddi, and this causes the entry of bile into the duodenum. And bile enters, it causes neutralization of acid, and it also causes emulsification of fat. So because here emulsification of fat is the major function of the bile. This is a very good example to see a cholecystograph, an X-ray image before and after a fatty meal. Look at this gallbladder before a fatty meal. So this is the radiographic image of a gallbladder before the fatty meal. After this, if a physician wants to study whether his gallbladder is functioning or not, what he will be doing is, first he will take an X-ray image, he will see the size of the gallbladder. After that, he will be asking the patient to have a fatty meal. Then after a few hours, another X-ray image is taken. So in this case, you can see gallbladder after a fatty meal. What do you see? You can see that the size of the gallbladder is reduced. See here, the size is this much. Here the size is reduced, which means that the gallbladder has been squeezed. Gallbladder contraction will take place after the fatty meal. Now you think about why the gallbladder contracts, because CCK is the hormone which is released, which will act upon the gallbladder, and gallbladder will squeeze and the bile will be released into the second part of duodenum as a result of fatty meal. So this proves that his gallbladder is functioning normally and his CCK levels are also normal. So this is usually done by the gastroenterologist just to make sure if his gallbladder is working or not, functioning or not. Chemical digestion of food. Let us see the starch, how it is digested, the carbohydrates, the polysaccharides. In the saliva, it has got the salivary amylase. In the pancreas, it has got the pancreatic amylase, which will break the starch into disaccharides. The carbohydrates are broken down into disaccharides, maltase, lactase, sucrase. Then it will be again broken down into glucose, galactose, and fructose. Whereas proteins will be acted upon by pepsin in the stomach, and there are pancreatic enzymes from the pancreas, the trypsinogen, chymotrypsinogen, which will cleave the protein peptides. Polypeptides will be acted upon by peptidases. They are the enzymes which will be acting on the polypeptides to break down into amino acids. Fats Unemulsified fat will be emulsified by the detergent action of the bile. The emulsified fat are with low size, smaller size, where the lipases can act. Then the pancreatic lipase will be acting upon, and it is converted into triglycerides. And triglycerides then form the fatty acids and glycerol. That's the basic steps involved in the chemical digestion of food in various parts of the gastrointestinal tract. Now let us see what are the major gastrointestinal hormones. Gastrin, which is released from the stomach. Protein in the stomach stimulates its release. Secretin, acid in the duodenum, stimulates its release. CCK or cholecystokinin, the chyme empties from the stomach. Fat and other nutrients enters the duodenum, causing the CCK release. GIP means it is gastrointestinal peptide. It stimulates the insulin release by the pancreas. We will be now looking at the major features of large intestine. The main function is the absorption of about 10% of the fluid. So this is the colon, which has got the ascending colon, the transverse colon, then the descending colon, and the S-shaped sigmoid colon. So these are the various parts of the colon: ascending colon, transverse colon, descending colon, and the sigmoid colon. Synthesis of vitamin K and B vitamins will be taking place in the large intestine, and that is the place where the formation of feces will be forming. That's towards the last part, because all the undigested materials will be entering this. And here you will be seeing a kind of movement which is called as a mass movement or a mass peristalsis. Waves of strong peristalsis participated by the entry of food in the stomach. It is called as a gastrocolic reflex. So this is a kind of reflex connection between the stomach and the colon. Whenever the food entry into the stomach takes place, you will be seeing that the patient or the subject will be feeling like defecating when the stomach is full. But this is very common in the case of young children or a newborn. This kind of reflex is more common. In the adult, it is not very common. You can see, but it is not very common in the adult. According to the various authors, they say that in the children, this gastrocolic reflex is commonly seen. That means when they eat something and if their stomach is full, they will feel like going to the washroom, so like for the defecation. So that is the finding. Usually the unwanted materials are the one which reaches the large intestine, which has to be excreted out in the form of feces. So it receives, the large intestine receives from the ileum and the ileocecal valve through the ileocecal valve. It also reabsorbs large amount of water, mineral salts, and some vitamins are also reabsorbed. There is also a microbial activity which is involved in the synthesis of vitamin K and folic acid. So this requires a friendly bacteria within the colon, the large intestine. And also some of the swallowed air which is there, it can also release like methane, hydrogen sulfide, etc. in the large intestine. Semi-solid consistency of the feces will be achieved, and the feces contain indigestible food products, mucus, and bacteria is delivered to the rectum by mass movement, mass peristalsis. So mass peristalsis is a powerful contraction which brings about the movement of the semi-solid, this waste materials and it is the feces, and it will be moving towards the rectum. That movement is called as a mass peristalsis. The defecation reflex. When the feces moves into the distended rectum, stimulation of stretch receptors will take place. You can see that these are the, here stretch receptors will be there on the wall of the sigmoid colon. Here you can see that that will be taking these pressure sensitive stimulus through the sensory afferent neuron. So you can see that this will be taking, right? So this is the afferent neuron that is taking this particular impulses. From here the stretch will be taken and it is going to the spinal cord. A spinal reflex is initiated in which the parasympathetic motor fibers stimulate contraction of the rectum and sigmoid colon and the relaxation of the internal anal sphincter. So here you can see where it synapses, and this synapse will be then connected to the nerve which is called as a pelvic nerve. So pelvic nerve, if you see, pelvic nerve is supplying to the entire part of the colon, of the sigmoid colon and the rectum, and it is also supplying to a structure which is called as an internal anal sphincter. So inside there is a sphincter which is internal anal sphincter, which is purely innervated by smooth muscle, right? The internal anal sphincter is here, so that is innervated by the smooth muscle, and this is the parasympathetic nerve. It is the pelvic nerve it is supplying. So as a result of stretching, by the fecal material will come here, it will cause stretching, and this stretching is sensed by the stretch receptors. Afferent information is taken from here through these afferent nerves, then through the pelvic nerve it will be coming, and then contractions will be taking place here, right? Contraction of the rectum and sigmoid colon will take place at the same time. Relaxation of the internal anal sphincter, which is the smooth muscle. Now the defecation will occur only if the external anal sphincter opens or relax, right? This is the external anal sphincter, which is a skeletal muscle. Look at this. This is the major difference. Internal anal sphincter is made up of smooth muscle, which is under the parasympathetic control, whereas external anal sphincter, this one, the outermost region, this is under the influence of, it is made up of skeletal muscle. This is smooth muscle, this is skeletal muscle. So smooth muscle will be always innervated by the autonomic nervous system. So here this is the parasympathetic nervous system, which is the pelvic nerve, whereas this particular nerve, it is a nerve which is called as a pudendal nerve. Pudendal nerve. So it is a motor nerve or a somatic nerve which is supplying. It is here. So this is an example for a somatic nerve. So it is a voluntary nerve, right? Because it is impulses is coming from the cerebral cortex, right? And then when the situations are favorable for this individual to defecate, then the impulses will be coming through the pudendal nerve, which is a somatic nerve, and it is supplying to the external anal sphincter. So external anal sphincter has to relax for this fecal material to go out, that is defecation to achieve. Otherwise what will happen? Usually the external anal sphincter will be constricted, so that even if this squeezes, the defecation is not possible. So that depends upon the situation. The situation, if it is favorable when the person is in the washroom, then definitely the impulses will be coming from the higher centers through the cerebral cortex. Then it will stimulate this pudendal nerve, which is a somatic nerve. And remember, this is a skeletal muscle, that is the external anal sphincter has to relax, and then the process of defecation will be taking place. So in this particular flow chart, you need to remember that the internal anal sphincter is innervated by smooth muscle, whereas external anal sphincter is innervated by skeletal muscle. And you need to know what is the function of pudendal nerve, which is a somatic nerve, and what is the function of pelvic nerve, which is a parasympathetic nerve. So this flow chart you need to just see and study from the various steps which is already mentioned here. This one, third one, see, if it is convenient to defecate, voluntary motor neurons are inhibited. That means this is inhibited, allowing the external anal sphincters to relax. So this will relax, and when this relaxes, easily the fecal material can be defecated, so that feces may pass out. So that depends upon a voluntary involvement. So that is under the control of the cerebral cortex. That's all about this particular lecture, and these are the references, and thank you for listening. We will be having again a session, a live session to clarify your doubts, and let us see you on that day. Thank you.